Good morning everyone. In today's video, I'll be talking about stationary phase. This is the third important phase of bacterial growth curve. The first one I have already mentioned, that is the lag phase. Second one is the log or exponential phase, where the bacterial cell that will divide at maximum rate possible for them. Now this stationary phase, how it looks like. Okay, this is lag, this is log, this is the stationary phase. This one, the phase which becomes horizontal to the x-axis. Okay, this phase become horizontal to the x-axis, means here the growth and death of the bacterial cell population remains constant, or there is a balance between the growth and death of the cell in a batch culture. Why I am saying this? Because in case of a batch culture, remember one thing, batch culture means closed culture vessel with single batch media. So we cannot add extra media once the cell attend the stationary phase. No, it's not like that. At the initial phase, we made the media. We have to make the media and then add the bacterial culture into that for their growth. So now there are generally two most important reasons why the bacterial population reach the stationary phase. Number one, there is a nutrient limitation occurs. Because at exponential phase, the cells, they generally use maximum amount of nutrients to grow and divide at a maximum rate. So nutrient limitation obviously results from that. And second one is, in case of aerobic bacteria, they need oxygen. Aerobic bacteria, they need always oxygen. So when too much bacterial cell are present in a batch culture, oxygen depletion also occurs. So without the oxygen or very less amount of oxygen, aerobic bacteria cannot grow. So they stop their growth, or they stop or ceases their cell division, but they remain metabolically active. This is the beauty of the stationary phase. Though the bacterial cells are not dividing, but they remain metabolically active. Okay, so this is the beauty of stationary phase. The bacterial cell in a closed culture vessel with a single batch media, which is basically a batch culture. The cells are not dividing in the stationary phase, but they remain metabolically active. And if few cells die, then there is a balance between the cell growth and cell death. So this is the stationary phase. Now there are few proteins which will help to survive this nutrient limited starvation condition of the bacteria in their media. One such enzyme is known as RPOS. This is basically the important component of RNA polymerase holoenzyme. Okay. Now, what is the function of this RPOS? These type of enzyme usually binds at a specific genes in DNA and then starts transcription of that specific gene, so that the bacterial cell can get those proteins or enzyme which will help them to survive in the starved condition, means the nutrient limited, oxygen limited conditions. So these are the starvation proteins which will make the cells much more resistant to the damage by the starvation and other stressful conditions of their growth media. Another one that is known as DPS, that is DNA binding protein for starved cells or from starved cells, DPS. These are the DNA binding proteins. Now, what is the function of them? What is the function of this kind of a protein? They usually binds with DNA and protect them from starvation during stationary phase. So this is the basic of the stationary phase, where there is a balance between the cell growth and cell death. Though the cells, they are not dividing actively, but they remain metabolically active, and there are enzymes like RPOS, TPS, which will protect the DNA and help to generate other proteins which will respond to the starvation process and make the cell much more resistant to the starved condition and other stressful conditions. Hope you like this video. If you really like it, please subscribe my channel. Thank you.